All right, so I hope you did that last task, and now we're going to talk about mitosis. In the next lesson, we're going to be comparing mitosis and meiosis, but for right now, we're just going to cover mitosis. Now remember, mitosis, I spelled that. Mitosis means my toes. Mitosis made my toes. After interphase, the cell goes into mitosis. We talked about that. The three reasons, and the only three reasons that a cell does mitosis, is to grow, develop, and repair. I'm going to highlight this word, if needed. The only reason we do mitosis is if we really need to. If we don't need to, we shouldn't do mitosis. This is when bad things start happening in our bodies. And those three reasons: grow, develop, repair. So for growth, right after two gametes came together to make all of the people in the world, it made a zygote. A zygote is only one cell. The human body is made up of trillions of cells. So that one cell grew through mitosis to create all the trillions of cells that make the person living today. Development. Your toes. Does a zygote have toes? No. So that zygote had to develop into an embryo and then a fetus, and it had to develop its toes. So development through mitosis. And the last one is repair. Have you ever fallen down and skinned your knee, or accidentally hurt yourself some other way? Well, when your body gets hurt, it has to repair itself. And it repairs itself through using mitosis. Growth, development, repair. There are 4 phases to mitosis: prophase, metaphase, anaphase, and telophase. And then cytokinesis is always going to follow after telophase. Prophase. Pro, I think previous, it's the first phase. Prophase is the first phase of mitosis. And there are 5 important milestones or things that happen here. I call them milestones because they're easy to keep track of. Kind of like a big milestone in your life is walking or talking. It's a big milestone that usually your parents remember. The first milestone is chromatin condenses into chromosomes. You're probably like, what is chromatin? Well, let's look real quick. So when you have a cell and you have your nucleus, the nucleus contains the genetic information. Well, in interphase, this genetic information is free-floating loosey-goosey chromatin. Chromatin is the free-floating DNA of the cell. When you get to prophase, that chromatin or free-floating genetic information condenses into chromosomes. Here are the chromosomes. We all know them. So the chromatin is going to condense. That means it's going to wrap really, really tightly around proteins. The reason it does this is because when it's free-floating, like a string in the wind, it can easily be injured, which is why it has to be protected and kept in the nucleus. Well, when we get ready to do our mitosis, we lose our nuclear membrane. It breaks down. You can see that because this membrane is starting to break down. Well, if the nuclear membrane breaks down, there's nothing to protect our chromatin. So we have to wrap it up really tightly, like string around a thimble, in order to protect it so it doesn't get injured during mitosis. The nuclear membrane has to break down so that we can move our chromosomes around and separate them evenly to get 2 new cells. The centrals, if you'll remember from structures and functions of the cell, these are centrioles. They're going to migrate to opposite poles of the cell. Poles here meaning sides. Like we have a north pole and a south pole on the Earth. Here we're going to have a left and a right pole, or a left and a right side of the cell. There's a very important reason it does that. Your centromeres, remember that that's the part of the chromosome that holds the two sister chromatids together. They're going to migrate to the center of the cell in this area. And then microtubules are going to form. Microtubule is just a more advanced term for these spindle fibers. Spindle fibers, microtubules. Your microtubules come from your centrioles, and your centrioles are on 2 opposite poles of the cell. That's very important, and we're going to talk about why when we get to anaphase. The next phase is metaphase. Remember, metaphase is for the middle. The 2 milestones here is that the chromosomes line up in the middle of the cell, like a conga line. They meet in the middle. So metaphase, you're meeting in the middle. Then your second milestone is that these microtubules, or your spindle fibers, are attaching to the centromeres. If you look closely, this one coming from the right is going to attach to that centromere there. This coming from the left is going to attach to the left side of the centromere. So we have 1 microtubule on the left, 1 microtubule on the right. That's important because now we're going to move into anaphase. And what happens in anaphase? A is for away. We are going to split our sister chromatids and move them away from one another. The 2 sister chromatids pull apart towards the centriole. The reason that they're being pulled apart towards the centrioles is because the microtubules are pulling them in opposite directions. So here's our chromosome again. We had a microtubule from this centriole connected to this part of the centromere. I'm sorry. I got distracted there by the dog. All right. And then on this side, we had a microtubule connect to this centromere. They are pulling away from one another and toward their own centriole. This is what divides the DNA. And then we have telophase. Telophase is the final phase of mitosis. There are 3 milestones for this phase. The first one is that the chromatids are at opposite poles. They're in opposite sides. And their nuclear membrane is reforming. Let's put a re right there, reforming. If you look closely right here, you can see the nuclear membrane that's coming back. This is important because we want that nuclear membrane there to protect our genetic material. Because our other milestone is that the chromatids are going to decondense back into chromatin. So once we have the nuclear membrane back, we're going to decondense into our free-floating genetic information, or our chromatin. This is the relaxed stage, and it's easier to do stuff with. And then, if you notice, we don't have our microtubules anymore. They have fallen apart, or depolymerized. All right, they've fallen apart. That leads us into cytokinesis. So cytokinesis is not a phase in mitosis. This is happening after, and it is where the cytoplasm divides to make two individual cells. The process is going to be different for plant and animal cells. For animal cells, we start out with that image that we had in telophase, right? And there is going to be a furrow that forms. It looks kind of like this. If you think of it, the bottom of a peach, it looks like the bottom of a peach. And what happens is that the cytoskeleton inside of the cell is going to attach to the cell membrane, and it's going to pull it, kind of like a drawstring bag, and it's just going to pull it. Well, it pulls it, and pulls it, and pulls it, and then when it gets small enough, the amount of cytoplasm connecting these two, it does a pinch to pinch it close and pull apart. And that is when we have the separate cells right here. We've got a membrane on each side. Plant cells can't do that. Why can't a plant cell pinch closed? Because it has a cell wall. It can't pinch closed because it has a cell wall, and cell walls are rigid. Therefore, they have to do something different. Here, a plate forms in the middle of the cell, and it will grow horizontally and vertically until it has filled the entire space. Once it has finished, it will completely separate the two cells, and you have two new cells. Okay, now identify each phase of the cell and tell me what it is. This is going to be 1, 2, 3, 4, and 5. Go ahead, hit pause, figure those out. Alrighty, so number 1, that's telophase. You can see here, you've got the genetic information on the two sides of the cell, and now it is starting to divide. Good job. Number 2, that's anaphase. You can see how the two sister chromatids have been pulled away from one another. They're being pulled away. Number 3 is interphase. If you look closely, you can see where the nucleus is, and then you have all these other organelles. See those other organelles? That's interphase. The cell is just doing what the cell does. I'll erase that so you can see it more clearly. All right, number 4, prophase. So if you look, your genetic material, it is starting to, it has condensed into the chromosomes, and the nuclear membrane has dissolved, or it's broken down. Now you have those chromosomes that are kind of scattered in the cell and are starting to concentrate in the center. And number 5, metaphase. You can see a line in the center of the cell where all those centromeres are lining up. They're meeting in the middle. Good job. All right, if I ask you, what's happening there? What would you say? Anaphase, right? Here, your two sister chromatids are being pulled away from one another. What would this one be? Metaphase, right? They've met in the middle. All right, I'm going to stop my questioning.